So in this applied biochemistry activity, since we're on the block talking about enzymes, all of the sort of applied biochemistry topics will be related to enzymes in some way. And so the first topic I'd like to talk about is a disease. It's an inherited disease. It's got the strange name of maple syrup urine disease. So it's got a strange name, but it's a very, very serious disease. So this is caused by mutations in the branched chain alpha keto acid dehydrogenase complex, which is a complex of several different enzymes, specifically 3 catalytic sites. And so what this enzyme usually does when it's functioning normal is it catalyzes the catabolism or the breakdown of branched chain amino acids. So that's leucine, isoleucine, and valine as shown here. So they're branched chain amino acids, of course, because they all have these hydrophobic branched hydrocarbon chains right here. So these are the 3 branched chain amino acids, or BCAAs. So that causes huge problems. So if you can't break down branched chain amino acids, what's going to happen is they're going to build up in your body. So this is normally noticed in infants. So infants can inherit this disease. So a mutated branched chain alpha keto acid dehydrogenase complex. And it's usually noticed because infants will have urine that smells like maple syrup. Actually, the earwax of these infants will smell like maple syrup too, which is really weird. And that just comes from this sort of buildup of branched chain amino acids. So in cultures that aren't really familiar with maple syrup, it's fenugreek, also can be described as a fenugreek smell. And so it's got a weird name, but this disease is really, really bad. So it can be fatal if untreated. So it will result in neurologic and respiratory failure from not being able to break down these branched chain amino acids. So it's pretty rare in the general population. So about 1 in 185,000 infants are affected. However, some communities where there's what would be called co-sanguineous like relationships or mating. So people who they'll have kids, but they're sort of, it's a small community, so the likelihood that you're somewhat related to the person you're having kids with, that can cause this disease to be a bit higher. So in some communities like the Amish or Mennonite community, this disease is more common. So in the Amish community, it's estimated as 1 in 176 infants are born with this disease. So this can be treated with a very, very strict diet. So you'd have very low amounts of branched chain amino acids in your diet, and then you'd have to make sure that you received enough other nutrients so you don't develop a nutrient deficiency. It can also be treated with like removal of the branched chain amino acids from the blood, basically through dialysis. And it can also be like fully reversed through a liver transplant. So the liver, when you're receiving the transplant, the liver's able, from the transplanted liver would contain a normal branched chain alpha keto acid dehydrogenase complex. So then that would basically cure you of this disease. So this complex right here is actually quite similar to the pyruvate dehydrogenase complex, which we're going to spend pretty much basically a whole lecture on. And then also the alpha ketoglutarate dehydrogenase complex, which is in the citric acid cycle, which we'll mention as well. So maple syrup urine disease, it's a breakdown of an enzyme complex resulting in the buildup of branched chain fatty amino acids. Now, the next disease we'll talk about is called Hunter syndrome. So Hunter syndrome arises from when you have a mutation in an enzyme called iduronate 2 sulfatase. So certain mutations in this specific enzyme, which is shown right here, will result in Hunter syndrome. And so what iduronate 2 sulfatase does is it breaks down glycosaminoglycans, or GAGs. So specifically, so we don't talk too much about these in the course, but they're basically carbohydrate molecules. So they're shown right here. They're carbohydrate molecules that have these sulfate groups on them that carry very negative charges on them, and they can make very long chains. 2 common ones are heparin sulfate and dermatan sulfate. So what these chains are is they'll be added to proteins on the cell membrane. And so because they carry that negative charge, they're very sticky, basically. And so they'll interact with a lot of different molecules, and they're really important in the extracellular matrix. So there'll be like a matrix of these glycosaminoglycans that's related to connective tissues. So when Hunter syndrome, this enzyme is not functioning properly and it's not able to break these glycosaminoglycans down. So again, they'll build up in cells. So they'll actually build up and can't be degraded. And this will cause a lot of problems all over the body, especially with connective tissues. So there's a lot of abnormalities in organs, including thickening of heart valves and the airway, which can cause a lot of problems. There's also physical sort of deformities, like sort of large head and very specific sort of physical features. And it can also cause neurological issues. So most patients who have this disease, it sort of varies as to how bad the disease will be. But if you have somewhat severe disease, these patients will die between the age of 15 or 20. So it's a really devastating disease. So this is actually an X-linked genetic disease. So this enzyme is on the X chromosome. So it has an X-linked recessive inheritance, and it mostly affects, I think it basically only affects males. So if you have an unaffected father, but a carrier mother who doesn't have the disease, but she did inherit the disease locus on that gene, she cannot pass it on to her daughters. The daughters can be carriers, but basically if a son inherits the X chromosome from the affected mother, that's the only copy of the X chromosome. So the Y chromosome doesn't have this gene on it. And so it's considered an X-linked genetic disease. It's also in the family of diseases called lysosomal storage disease. So lysosomes are organelles within the cell that are responsible for breaking down raw materials. So basically, the glycosaminoglycans they'll try to be broken down in the lysosome, but they can't because this enzyme is not present. So there really are limited treatment options for this disease, but it is a very good candidate for gene therapy. So as things like CRISPR take off in the next few years, and we start using that to change the genes in human beings to treat different genetic disorders, you could imagine how you could use that to great effect by just correcting that mutation on the specific gene that causes Hunter syndrome. Now, a similar lysosomal storage disease is called Gaucher's disease. And so that is caused by mutations in an enzyme called glucocerebrosidase. And so that enzyme is shown right here, and its function in the cell is to catabolize glucocerebroside. So these are a type of membrane sphingolipid. So we'll talk about those in the lipid lecture here in the next block. And so it's also a lysosomal storage disease because it's the same kind of situation. So what glucocerebrosidase does is it's supposed to break down these glucocerebrosides. So a glucocerebroside is right here. So it's a sphingolipid. So this is an example of one. So it'll have a glucose or other groups on it. Then it'll have a fatty acid with a variable chain length. And then I'll have this specific group here in red, which is sphingosine, which is what makes it a sphingolipid. And so this is an important lipid in the cell membrane. When this enzyme here is mutated, you can't break this down. So this reaction right here cannot occur. And so the same thing as with glycosaminoglycans will happen. These glucocerebrosides will build up actually in red and white blood cells. And so when those red and white blood cells need to be degraded by these immune cells called macrophages, so they will take those molecules to the lysozyme. But since the enzyme isn't working, it'll sort of just build up in the macrophages, and that'll continue to happen until you get these macrophages that are really weird looking and will build up into cells. And so the outcomes for Gaucher's disease really vary by patient. So there's a few different types depending on how much enzyme activity you have, but it often can cause very serious problems, including a very enlarged liver or spleen. So often people with Gaucher's disease have a spleen that weighs about 2,000 grams when it should weigh 200 grams, so 10 times as big. There's also liver and skeletal disorders, bone lesions, lymph node swelling, and neurological complications. So it's a really, really bad disease as well. Again, like we saw with the other disease, there's higher incidence in specific communities that where people may be sort of related more than just in the general population. So there's a way higher incidence. So like 100, you're 100 times more likely to get Gaucher's disease if you are an Ashkenazi Jewish person. And so again, the outcomes for each of these patients with Gaucher's disease can vary, but this disease can be treated with enzyme therapy. So you'd basically get the enzyme into your body so it can break down those glucocerebrosides. Unfortunately, this is incredibly expensive. It costs about $200,000 per patient per year to do this type of treatment. So it's that will add up. So that's not obviously the best way to treat it, but it does work and can help the outcome of the patient. You could also treat it with some drugs that are coming on the market that would actually lower production of glucocerebrosides. So there'll be less buildup if you produce less of these molecules. And again, this would be a very good candidate for something like gene therapy using CRISPR therapy. So that's Gaucher's disease. It's a mutation in the enzyme glucocerebrosidase, and it's also a lysosomal storage disease where individuals cannot break down these lipids called glucocerebrosides. So now we can move on to some syndromes that are a little less serious but still involve enzymes, and one of those is lactose intolerance. So there's an enzyme called lactase that is basically expressed in the small intestine of human infants, but most people, as they grow up, basically lose that lactase activity on aging. So what lactase does is it breaks down this carbohydrate molecule called lactose. So lactose is formed from glucose and galactose. So your body can metabolize, obviously, glucose, and it can metabolize galactose individually, but it can't, it first has to basically break this bond here to release those two molecules. And that's what lactase does. So it'll break that bond between glucose and galactose. So that's really important as an infant so you could digest milk from your mother. However, most people, so about 65 to 70% of people on the planet, will actually lose that lactase activity in the small intestine after they age. So they are now unable to convert lactose to glucose and galactose, and they've developed what's called a lactose intolerance. So what happens is basically a bunch of digestive issues. It can cause, sort of upset stomach and stuff like that. It can be more or less serious depending on how much residual lactase activity that you have. However, over time, certain populations have maintained lactase activity into adulthood, and that's sort of corresponding with the importance of milk, the domestication of cows, and the importance of milk in the diet. So you can see that certain, mostly European populations, some African populations, and some Middle Eastern populations have all have varying levels of retaining this lactase activity into adulthood. So this disease can usually be managed just by avoiding foods containing lactose, so avoiding milk. Things like cheese generally have less lactose in those, so those are sort of those are sort of more tolerated in general, although it varies between people. And you can also take lactase supplements. So right here is an example, Lactaid. And so this is, you can see right here, it's a lactase enzyme supplement. So it'll come in a pill that basically includes the enzyme in a dried form. And when you swallow it, it'll basically break down the milk in your stomach so you can deal with that. So lactose intolerance, that's caused by a lack of lactase in adult individuals who have lost the ability to break down lactose. Another less serious issue involving enzymes is alcohol insensitivity. So there are two enzymes involved in breaking down alcohol, so ethanol that you would get from beer, wine, or spirits, into a molecule that you can sort of deal with, like acetone. And so these enzymes are alcohol dehydrogenase and aldehyde dehydrogenase. So Alcohol dehydrogenase is involved in this reaction right here. So the conversion of ethanol. So it's it's an oxidation of ethanol to acetaldehyde, and then you reduce NAD plus to NADH. And then that acetaldehyde, which is shown right here, is going to be further reduced to this molecule here, which is acetone. And so that's performed by aldehyde dehydrogenase. And it's also an oxidation reduction reaction. This molecule, acetaldehyde, is oxidized to acetone, and then NAD plus is reduced to NADH. And so in your body, there's actually two forms of aldehyde dehydrogenase, that second enzyme. So there's a cytoplasmic form with a very high Km, which means for it to get half its maximal activity, you kind of have to have a high amount of acetaldehyde present. And there's also a mitochondrial form with a low Km. So that one is going to be very active even when there's only a low amount of acetaldehyde present. So some people have a mutation, actually a single point mutation, in that low Km mitochondrial form of aldehyde dehydrogenase, which is going to basically lower its activity. So what that means is that these individuals basically have an alcohol insensitivity. So if they drink alcohol, it immediately results in facial flushing and tachycardia, which is like a fast, rapid heartbeat, even after just a few sips of alcohol. So there's certain populations on the planet where this is very common, especially some East Asian populations. And then there's some individuals who are basically just don't have aldehyde dehydrogenase active at all. And they're basically, you could call them allergic to alcohol, even though that's not exactly the right term. So this buildup of acetaldehyde has actually been linked to hangovers. So if you've ever experienced a hangover, a lot of research suggests that that's a buildup of acetaldehyde that your body sort of can't keep up with. And sort of those negative symptoms are associated with that. So people who can, drink a lot and basically have, hollow legs, have, very high activities of this alcohol dehydrogenase and, more importantly, aldehyde dehydrogenase. So that's alcohol insensitivity involved with two enzymes that are responsible for breaking down alcohol from alcoholic beverages. So since we're talking about enzymes, we can also talk about poisons or toxins that actually can really cause a lot of problems if you're exposed to them, because they inhibit enzymes that are really important for the function of your body. So one of these is alpha ammonidin poisoning. So alpha ammonidin is this molecule right here. So it's actually a cyclic 8 peptide molecule. So this is a polypeptide here, and it forms a circle. And it's a molecule that's produced by this mushroom right here called the death cap mushroom, or Amanita phalloides. So Amanita is a big, there's a lot of different mushrooms in the genus of Amanita. Amanita muscarium, you've probably seen before. That's sort of a Super Mario Brothers looking mushroom. Some people take it recreationally, even though they really shouldn't. But these Amanita mushrooms are responsible for 95% of the poisonings of mushroom poisoning. So if this death cap mushroom is present in an area, a lot of times there'll be signs like this saying, do not eat any of the mushrooms in this area. And that's because this molecule, alpha amanitum, is a really, really potent inhibitor of this enzyme right here called RNA polymerase II. So RNA polymerase II is very essential for the functioning of your cells because it transcribes mRNA from DNA. So it's this first part of the central dogma, taking DNA and making mRNA out of it. So we'll go through that in block 5. So if you aren't making any mRNA, you aren't making any proteins, and basically your body is not going to function anymore. So if you accidentally ingest one of these death cap mushrooms, you won't see any symptoms for a little while. So it'll inhibit your transcriptional machinery, but you have enough proteins in your body to sort of function kind of normally. And that's actually really bad because you don't know anything's happening and you can't get your stomach pumped or anything like that. So after about 24 or 48 hours, you might showing some symptoms, But basically, your body will basically start to shut down because it's not making any new proteins. And so it'll cause death usually about a week from kidney and liver failure. So you just don't have enough new proteins to keep up with the kidney function and your liver function in your body. Now, this alpha amanita molecule is actually really important for biochemistry as well because it allows you to distinguish RNA polymerase 2 from another RNA polymerase 1, and RNA polymerase 3. So it's a really potent inhibitor of RNA polymerase 2, which makes mRNA, but it's less of an inhibitor of, I think it's RNA polymerase 3, and then it does not inhibit RNA polymerase 1. And those are responsible for making rRNA, which is RNA that gets made into ribosomes, which we'll talk about in a second, and then tRNAs, which are also involved in translation and really important RNA molecules. So that's alpha amanita poisoning. It's a toxin made by mushrooms, the death cap mushroom, and it is a potent inhibitor of the very important enzyme RNA polymerase 2. And then lastly, we'll talk about ricin poisoning. So this is actually an example of where an enzyme is an inhibitor of another enzyme, or something basically like an enzyme or a molecule with enzymatic activity. So ricin is actually a protein molecule right here, and it's an enzyme, and it's a potent toxin that's produced by castor beans. And so these are just beans, and what it does is it cleaves glycosidic bonds in the ribosome. So the ribosome is a really interesting molecule. It's very, very important. We'll talk about it in block 5. So it's involved in translation. So we saw how RNA polymerase 2 is involved in transcription, so making mRNA from DNA. So the ribosome takes that mRNA molecule right here, and then using those tRNAs, it will produce a protein chain based on the sequence in mRNA. So we'll spend a whole lecture talking about this process. And so what ricin will do is it'll basically chew up that ribosome, because the ribosome's actually, it's a catalytic enzyme, but it's made of a combination of rRNA and proteins. And so it'll actually chew those ribose groups off of the rRNA, and it'll shut down the ribosome. And so this is a highly, highly potent toxin. So one ricin protein is capable of completely shutting down and destroying a cell. So this certain poison has been used extensively in fiction. So there was a running story in Breaking Bad where Walter White isolated ricin from castor beans and then was going to use it to, murder people who were in his way. And so he had a cigarette that was basically had the ricin in it. So ricin's more toxic if you inhale it or if it's injected into you, rather than ingested. So ricin was actually used by the KGB to assassinate this Bulgarian dissident named Georgy Markov. And the KGB delivered it by using a modified umbrella tip where they injected ricin into this individual, and it actually killed him. So ricin is an extremely potent toxin produced by castor beans that it's an enzyme itself, but it also destroys the ribozyme catalytic enzyme of the ribosome that's involved in transcriptional machinery.